Okay, folks. As I stated in the announcements, my old recordings of this subject matter, they went in overtime because ordinarily when we do this class synchronously, where everyone's required to attend the Zoom meetings, I've been known to go the full 4 hours. And when we combine lecture and lab and the fact that I like to walk everybody through everything, just as we would do if we were meeting in real life and meet space, not virtually, those videos were just too much for the software, the platform to handle, and they wouldn't allow me to upload them. So from here on out, I'm going to be doing small bite-sized videos. I try to keep them. I really do not like video more than 40 minutes. That being said, there are some subjects where we may be unable to avoid that. Right now, this minute is one of those. So what I'm going to do is immediately cut right to the chase here. This is module number 3. Well, lecture number 3, really. We had 2 lectures, terms of direction, body regions. Now we're going to have 3 and 4, which are combined into like module 2 here. Cell structure, eukaryotic cell structure, and then cell division. So let's go ahead and get right to it right now this minute. Let me share the screen with today's regularly scheduled PowerPoint, or since this is an asynchronous course that I'm making this video for, technically it's tomorrow's PowerPoint, so you should be viewing this stuff on Wednesday, June 16th. And like I mentioned, you don't want to view too much of this stuff all at once because it's overload, but you do want to view it at, don't procrastinate. So cell structure. Let me fix this PowerPoint so you, the viewer, depending on if you're viewing it on a smaller Chromebook or something with a smaller module, you'll have an easier time viewing it. And let me just pin my video here. I'm trying to shrink my Zoom toolbar down because it's interfering and I cannot do it. Oh, well. Oh, snap. I just lost it. So hold on. I have video problems here. Here we go. Let's get this and shrink this window. Okay, so here we are. This is about as good an image as I'm going to get. So cell structure. We humans and anything else in the animal kingdom, we're made of a certain type of cell. And these are called eukaryotic cells. There's another type of cell called prokaryotic cells. You'll cover those in microbiology class. Eukaryotic cells are big and complicated compared to prokaryotic cells. If you had to think of it this way, think of it as this way. A prokaryotic cell is like an old 1960s pickup truck or muscle car, an old Ford Mustang, Falcon, Chevy Nova. I have a Dodge D100 that I'm semi-restoring, like I haven't done a thing on it in 6 years since my kid was born, but you know how it is. Old cars are simple cars, very few things in them when you think about it. That's like a prokaryotic cell. A eukaryotic cell is like the latest, greatest state-of-the-art Cadillac, and the latest Toyota, the latest Escalade, the latest Lexus, Infiniti, super complicated vehicle. Even if it's not the luxury model, even if it's the base model, these cars have so many features, there's so much complexity, so much things to go wrong and break on them, and it's insane. Well, that's how a eukaryotic cell is. Eukaryotic cells, like modern cars, are way more feature loaded and way more efficient than the prokaryotic cells that you study in microbiology class. Now, one of the nice things about eukaryotic cells, by virtue of their complexity, they can cooperate, work together, form teams. So eukaryotic cells can work together as a team, like the Dodgers, like the Lakers, like the Rams, like the Chargers, like the Galaxy, like LA Football Club. They were the Kings. They work together as a beautiful team, and this team creates what we call a multicellular organism. Prokaryotic cell. There's just like some dude in a basketball court after school. Everyone else is horsing around. He's practicing his layups, shooting baskets, whatever, dribbling. Whereas multicellular organisms, they're like a professional, a major league team. They're skilled. They're really good at it. If you were to look at yourself in a mirror and think of yourself as a team of individual players, baseball team, football team, basketball team, hockey, not that many players on the court at any one time, might be more in reserve, more sitting in the dugout and a bunch laid up with medical issues. but your team is 50 to 100 trillion. That's 10 with like, actually a trillion is one with 12 zeros after it. That is a big freaking huge number. We're depending on which textbook you read, which authority you consult with, we're made of 50 to 100 trillion cells. That is a big team with a lot of players. Each one of them cooperates like an all-star. Okay, so like I said, you'll study prokaryotic critters, bacteria in microbiology class, and I'm not saying anything more about it. I will say this about microbiology. In the lab, if you're taking the class in person, follow directions like your life depends on it in the microbiology lab, or that course will end you. Very unforgiving in terms of not getting the right results in lab class. That's all I'm saying about these now. Let's get back to our proper general human anatomy material. So eukaryotic cells, they're bigger. 10 to 100 ohms. Now look at this thing I'm highlighting with the cursor here, That stands for micron, 1/1,000,000th of a meter. So I'm gonna hold up my fingertips to the camera here. That's about 1 millimeter there. So a micron, I guess I can just stop share real quick here. So if this is like 1 millimeter there, a micron is 1/1000th of that. 1 micron truly is microscopic. A speck of dust, let's say you're dusting the house, vacuuming, and it kicks up that dust and the afternoon sun's coming through the window. Those tiny little specks of dust you see in the sun, insanely tiny, those are like 300 microns. So those are 300 times bigger than 1 micron. That is truly a tiny, tiny division of measurement there. So our cells, smaller than a speck of dust, most of them. There are some exceptions to that rule, though, that are big enough you need a ruler, even a yardstick to measure the length of some of them, but in terms of their cross-section, still microscopic. Okay, so eukaryotic cells, they're compartmentalized like a big 8-bedroom, 8-bathroom mansion somewhere, lots of rooms. Prokaryotic cells are like a single room occupancy, like a hotel room somewhere. You got the main room, maybe a little bathroom, kitchenette off to the side, but there's not a whole lot of internal compartmental space in a prokaryotic cell. And they're tiny little dudes, like 1 micron to 10 microns. So our cells that we're made out of, these are big, big cells. Okay, so this diagram here, I just like pulled that off the internet. That's copyright for Pearson. They're the ones that publish one of the textbook publishing companies we use. Okay, and as you know, for this class, I provide most all of the lecture material on my PowerPoint. So I'm not really supposed to say this, but in theory, you can totally ace the daylights out of this class and not even have to buy a textbook. The lab manual, lab material I provide for free anyway. So you're good to go there. If you have a question about a textbook, any old textbooks do, just to have something to use to refer to pictures and lists and names and stuff like that. Now, getting back to the subject matter here, the internal structures that give eukaryotic cells a complexity, these are called organelles. Now, that's just a kind of like a diminutive of the word organ. You think, oh, as humans, we have internal organs, heart, lung, liver, brain, kidneys, bones and muscles are organs. A nerve is an organ. But cells are tiny little doodads, and bazillions of those make up each organ, and yet they themselves have their own internal working parts we call organelles. Okay, not all of these organelles form compartments or spaces. Some form structure like a skeleton, and then some are involved in movement, some just form barriers, and some are just hard little balls of RNA and protein that make, protein molecules. Okay, the organelles that actually form separate little spaces like the rooms in a big house with lots of different rooms in it, I call these MBOs, which is short for membrane-bound organelle. Okay, so on this little TEM here, transmission electron micrograph of a liver cell, you see all these little circles and ovals and things, and even some of these little zigzaggy lines in this big dot, this big circle here. These are all MBOs, membrane-bound organelles. Now, if we zoom in here, dare I do this? I already messed with this PowerPoint file once accidentally. So I can go ahead and zoom in. That's the nice thing about having high-res images is that I can do this on my lecture presentations. That the downside, of course, is when you're trying to upload or download them from Canvas, it takes forever and it can cause your computer to crash. Anyway, or at very least cause PowerPoint to crash and have to restart, which it happens to me all the time. Anyway, you see these little zigzaggy lines there. That's a membrane-bound organelle called the rough endoplasmic reticulum. You see all this grainy stuff. It looks like sand, gravel, or salt and pepper, sprinkled on the image here. Those are not MBOs. Those are not membrane-bound organelles. Those would be other things. They can still be organelles, just not membrane-bound organelles. Okay, let me shrink this image back down here because if I don't, it will wreck the PowerPoint file. And let's go back. So if we were meeting in person, after this lecture, in a short break, we'd reconvene in the same room for lab. And in today's lab, we would, with a microscope, a toothpick, a microscope slide, a cover slip, and some methylene blue or iodine stain, we would take our own cheek cell scrapings from the inside of our mouths, deposit them on a glass microscope slide, put a droplet of stain on there, and then put the glass cover or plastic cover slip on there, chuck that bad boy on the microscope stage, and we would look at our cheek cell, our human epithelial cheek cell smears. Buccal epithelial cells from the buccal region are our cheek in between the upper and lower jaw inside our mouth. So on our specimens, our own wet mounts we would make with our very own cells in class in real time, we're guaranteed to see 4 things. And it really would be nice and awesome if we could see a 5th, but that doesn't always happen. We would see how big these cells are. We would see the cell membrane that's kind of like the cell skin, keeps the outsides outside of the cell and the insides inside of the cell. The cytoplasm, which is all that gooey stuff inside the cell, but not a part of the nucleus itself. And of course, the cell nucleus. There's a little itty bittier thing inside the cell nucleus, a little darker dot within that dark oval or circle called the nucleolus. Sometimes we can see that, sometimes we can't. It takes careful work, sometimes a fair amount of stain applied, not too much, not too little, in order to get a proper view of that. Think of it this way. Methylene blue, this is like a ghost pepper sauce or a Carolina Reaper sauce. A little bit goes a long way, but at the same time, you do have to use enough. Most folks end up gobbing it on there and using it too much of it, and our cells stain a little too dark, and then it's hard to differentiate that nucleolus from the rest of the nucleus. Okay, so that's one thing we would do in lab class. The other thing we would do is look at these very large plastic models that are about a foot across, 100, actually, what am I saying, 100? 30 centimeters is about 12 inches in diameter. And this particular image here, this is the older version from like the 1970s. We have some newer models, very recent, that show things a little different. Nonetheless, this image, my old cell phone, before I finally had to get rid of it and upgrade from years ago, shows a few important things. We have the cell membrane. That's like the cell skin, keeps the outsides outside, the insides inside. In other words, it regulates movement into the cell and out of the cell. And then we got corporate headquarters right here in the center, the cell nucleus. This directs the actions of everything else inside the cell to a limited degree. Some things act all by themselves and don't need much help from the nucleus, and other things are driven by external events outside the cell, what we would say extracellular stuff. Okay, so the cell nucleus, this contains what's called a genetic program. Everybody and everything that's alive has a genetic program. Think of your DNA, your genetic program, and all of your cells as the assembly, operating, and maintenance manual for a whole new you. It's embodied on a molecule called DNA, deoxyribonucleic acid, and it's here inside the nucleus. Okay, now getting back to what we would see with our cheek cells in class, this particular image you see here, the stain was applied very, very lightly. You'll learn to have a light touch if you've ever had ghost pepper sauce and you're putting it on stuff. And you're talking to a dude here that used to eat habaneros. I would eat habaneros in the old days on a dare. I don't know if I would do that with a botulachia or Carolina Reaper or Trinidad scorpion pepper. But, the really, really, really flaming thermonuclear warfare hot pepper sauces, you just use a tiny bit. The chemical dye we use for histology here, called methylene blue, same way. So this is a perfect slide you're looking at here, perfect image, just a little bit applied, not too much, not too little. And then we see everything. So what I have labeled here, number one, is light blue material here is a cell cytoplasm. That's basically everything inside the cell membrane, but it's not a part of the nucleus. And then number three here, we have the outside edge of the cell that's delineated by the cell membrane. You can also call cell membrane plasma membrane. And then this dark blue oval here is the cell nucleus. Now, what I'm going to do here is I'm going to expand this high-res image, blow it the heck way up, and try not to wreck my PowerPoint here. So this little nucleus here, I'm circling it with the cursor. You see that little dot right in the center? That's the nucleolus. If you think of the nucleus as corporate headquarters, the control center of the cell, the nucleolus, it's kind of like payroll, accounting, IT department. What good is a CEO going to be or a president going to be if the hired help doesn't get paid and the water and power and gas don't get paid? What good is the corporation or agency going to be if they don't have an IT department to manage everyone's computers and internet and keep that stuff working? So this little dark dot here, that keeps everything else in here organized and accessible. So a lot of textbooks will say the nucleolus, that little dark dot, it organizes the contents of the cell nucleus. Okay. And if you've ever been involved with running a business, especially a small business, it's okay. It's one thing to have the visionary, the CEO, and leadership, but they will get nowhere in this world without competent accounting, finance, payroll, shipping, and IT department to keep the gears turning in that organization. So our cells are the same way. Okay, now, and of course I mentioned it, this slide is pulled, oh no, this is frog skin epithelium. And this is, we'll talk more about this image Thursday when we cover beginning histology. This shows a tiny little nucleolus much, much more regularly, much better than a previous slide. The only thing I don't like about this slide is it's not from humans. This is from a frog, a non-human animal. And I always hate having to study and deal with non-human animals when we're talking about human anatomy and physiology. There are times when that's unavoidable, but I always give that the side eye. Anyway, this image, even though it's not from humans, does show the nucleolus of each cell beautifully. You can also think of a nucleolus the way I wrote it here. It's like the nucleus is a library of genetic information. The nucleolus is a librarian that controls what gets checked out, what gets put back, where it goes, things of that nature. Okay, now this here is here's the old model and here's the new model. You can see a few little differences in the shapes of the old versus the new. On the lab exams and quizzes, I just have some of these same photos here and I just Photoshop in some arrows and numbers and you have a question to answer and it'll typically be multiple drop down. There are a few assignments where you have to type in the answer and then I manually check those because the computer doesn't always check the spelling very well. But anyway, two different models of essentially the same thing. Now what these two models show is what we call the endomembrane system. So it's a membership, it's a club of organelles that are either directly physically touching each other or they maintain what's called vesicular contact. We'll talk about vesicles in a second. Just think of them as like trucks, and the organelles is like a warehouse, a factory, distribution center. You know, you have UPS, FedEx, DHL, Amazon trucks going from one distribution center to another, one warehouse, one facility to another. They're like transport vesicles, and the big 300,000 square foot facilities that they're going to and from, you know, those are like organelles. Okay, so we got a list of these: cell nucleus, cell membrane, those are parts of the endomembrane system. Then things I haven't mentioned yet: rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosomes, and the aforementioned transport vesicles, or just vesicles. Okay, so same images. I just use Photoshop to black everything else out so it's not distracting, and then we can just see the important things at hand. So RER, rough endoplasmic reticulum, physically three-dimensionally, it's like a stack of empanadas or pita bread. You know, you got a big disc-shaped piece of bread that's hollow on the inside. You can stuff it with good stuff. The hollow space on the inside of the RER, very technically, that's referred to as a cisternae. And with rough endoplasmic reticulum, you look at the surface, these little white dots on here on this model, those are called ribosomes. Ribosomes are organelles, but they're not membrane-bound organelles, and their job is to act like little protein factories. That's what they do. They make proteins all day long. So the surface of the rough endoplasmic reticulum has so many ribosomes, it looks like it's sandpaper. It has this textured, bumpy appearance. You can almost think of them as like poppy seeds on the surface of a poppy seed bun. By the way, a public service announcement: if you have to take a urine test to hold your job, say you're a crane operator at the port, you're running heavy equipment or something like that, avoid eating poppy seed buns because they will make you test positive for opiates and type compounds, because poppy seeds where we get opium from, morphine, codeine derivatives, and heroin, all that other stuff. Okay, the RER, in real life, IRL, is way more layered and extensive than the plastic models that we have would indicate. In order to replicate the level of complexity you see here on this microscope image on our plastic model, they'd be running $1,000 a pop instead of $430 a pop. And, you know, buying these anatomical models and stuff for our class, MSRP, manufacturer suggested retail, oh my God, that runs into some serious five-figure, six-figure dollar amounts there. So the plastic models oftentimes will oversimplify real life. And this is one case where they sort of do. Okay, so we have the rough ER here. It's like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 layers just in this little tiny part of the cell here. We have a cell nucleus. And the thing about the RER is it's always very intimately, tightly associated with the cell nucleus, like it's attached to it, like it's just stuck right on the outside of it. This image here is colorized from an ordinary black and white TEM, transmission electron micrograph. But it shows the cisterna on the insides, and you see this grainy lamp. I'm going to blow this up. Why? Because I can. Yeah, some of these images I have here are my own images. Others, like this one, are stock photographs from another library. But at any rate, you can see these little the graininess in here. That's the cisternae. Actually, I should say that's the ribosomes mounted on these things. This gray area on the inside would be the inside of the ribosome, inside of the RER proper. Okay, this big circle here is actually something else I haven't talked about yet. That's a mitochondrion. One angle, a long, narrow side view of it. Let's go ahead and put this back, or we should probably use a proper term, transverse view of a mitochondrion. Let's just put that back and go to the next one. Our models really don't show this next element that much, that extensively. SER, I call it, smooth endoplasmic reticulum. So SER is called smooth because it does not have ribosomes. So the surface is smooth rather than bumpy. If you wanted to get an idea of the three-dimensional space of the SER, in real life, the closest thing I've ever seen is the inside of Tom Sawyer's Island at Disneyland. Back in the old days when I was old enough and not in so much pain as I'm young enough, and I was not in so much pain and aches and stuff that I could crawl inside there. It's like this curved passage where it's all, there's not a straight line or a right angle inside there. It's all these curved passages that branch off and go every which way. You know, branch right and left, go up, go down, come back around, reconnect. Just this hollow, tubular, round network of passageways and spaces. Well, SER is a site of cell membrane synthesis. So all these MBOs, this is where their membrane, the M part, is manufactured. A rough endoplasmic reticulum is where proteins are made. Smooth endoplasmic reticulum is where cell membrane, phospholipid materials are made. And of course, we cover more about that in physiology class. But basically, the ingredients for building a human cell are all, I should say, the factories. The factories for building a human cell are located in the RER and SER. Okay, now, real world image of the SER, another transmission electron microscope. Cut through it. Look at all these passageways in here. This is a very nice artist's illustration here from a textbook. I didn't do this, someone else did. A lot of the images on these PowerPoints are mine. Some I just lift off the internet from various sources when I made these. Anyway, we got the nucleus, rough ER with the little ribosomes on there, and you can see the smooth ER. It does have a different three-dimensionality to it, a different 3D physical shape. And with this TEM here, transmission electron micrograph, I don't know if it's 11 bazillion and a half. It might just be 11 bazillion power magnification. No, just kidding. It's probably like, I don't know, 5,000 times magnification. That's a good guess. Maybe 7 to 10,000 times magnification. I'm not super sure. Actually, what am I saying? No, I'm looking at this bad boy. And I'm thinking maybe 20 to 30,000 times magnification. If the magnification's not shown on the image itself, then it's unknown. Anyway, you can see all these little passageways. It's just like they slice through it with a microscopic knife called a microtome. And this is what they see on the inside. Okay, Golgi apparatus. This here is the newer model here that shows the Golgi apparatus the way more contemporary textbooks have it illustrated. So Golgi apparatus, it's kind of like a fulfillment center. It doesn't really make stuff. It's more of a picking, packing, shipping, receiving plant or facility. So stuff is made elsewhere, like the smooth ER and the rough ER, and then it goes to the Golgi apparatus where it will be modified and packaged for shipment. Only thing is, cells are not Amazon or eBay sellers. They don't ship, they secrete. They release material outside of the cell membrane into the extracellular environment. Extracellular meaning the stuff outside. Okay, so when a cell releases material outside of itself, we call that exocytosis. Secretion is another term there. So let's say we're talking about the pancreas, which produces insulin and glucagon or pancreatic lipase. It secretes the cells inside there, just release this material outside of their own cell boundaries. Okay, so insulin goes into the bloodstream, but pancreatic lipase or sweat, those things will go just onto the surface of the body or inside of a duct that's not connected to the bloodstream. Okay, cells in a gland like the thyroid gland, the pancreas, the pituitary gland, they're going to have a really active endoplasmic reticulum, rough ER, and their Golgi apparatus, like an Amazon facility. Everybody's going to be working some serious overtime there. Okay. So here's a very nice colorized image of the Golgi apparatus. Again, I'm looking at this thing and I'm guessing several tens of thousands of power, at least 20 or 30,000 power minimum magnification. Again, artificially colorized, but you can totally see all these layers here. The way this works is receiving is on one side, where material produced by the endoplasmic reticulum, the vesicles dock here, and then it works its way through as it gets modified. And then shipping is on the other side, where vesicles take it to the cell membrane for secretion or exocytosis. Okay, so again, right here circled in pink would be, or where I'm highlighting it with the cursor in pink, that would be where Golgi apparatus would be. Now, lysosomes, they're little round nondescript suckers. They look like a lot of other things. Here's the problem. The plastic model answer key, I believe, gives us the yellow ones with the orange-red centers are labeled lysosomes, and these gray ones with the purple centers are something else entirely. So lysosomes, their main job, we were told, is cellular digestion and recycling of old worn out materials in the cell. They have an internal pH of 3.5 to 4.5, which is pretty darned acidic. Not battery acid, stomach acid levels of acidity, but like glacial acetic acid, enough to corrode and damage metal and blister the skin. So digestive enzymes that break down proteins work very well at this pH. So for this reason, they've been called cell stomachs. When I was a little kid growing up, taking these classes in high school and undergrad and college, we just used the term cell stomachs for lysosome. Other people have called them suicide bags because it was thought at the time that they were important in something called apoptosis, a pre-programmed cell death. Oftentimes, cells will commit suicide. They'll just break apart from the inside out. A cell that's hopelessly overrun by viruses may do this. We were taught that, say, little tadpoles in a frog, a baby frog looks like a fish, of course. It doesn't even have the legs, it just has a tail and a big head. Humans, when we're at certain ages of stages of development, before we're even born, will have tails too. A lot of these structures, we were taught that the cells making those things up would undergo apoptosis and the nutrients reabsorbed for use elsewhere in that little developing organism. Okay, so that's that for lysosomes. Now, two other things that I should say, the next two items are not a part of the endomembrane system anymore. So we're done with the endomembrane system. We finished that at lysosome. Mitochondria are membrane-bound organelles, and mitochondria are like cell power plants. They take two things in this world. One of those things you eat, food molecules, and the other thing you breathe, oxygen from the air. Now they're going to take those food molecules, so you have yourself a bacon, double cheeseburger burger. You're going to have carbohydrates in a bun. You're going to have proteins, amino acids in the patty and the bacon, lean part of the bacon. And in the cheese, you'll have some more proteins. And then in the fat part of the hamburger and then the bacon fat, you're going to have what are called lipids. All of those big molecules that makes up food, they get digested down in the stomach and small intestine and absorbed. And ultimately, what you get at your individual cells here are small individual molecules of glucose, ketone bodies, from lipids, things like that, and individual amino acids. So mitochondria are basically going to take glucose, sugar, and oxygen, and what they're going to do is harvest the energy by combining those two. So you can say they're energy transducing organelles, cell power plants. They make a chemical called ATP, adenosine triphosphate. Well, I talk a lot more about adenosine triphosphate in physiology class. And if you're taking this course at LA Southwest College, chances are they may have required that you take Bio 3, non-majors, actually Bio 3, Bio 5, Bio 10 also as a prereq. And in those classes, you'll have covered this as well, cell respiration. So a lot of mitochondria. In a cell that you might find in, say, the liver, you'll see thousands of these per cell. Other cells, they only have a few hundred. Now, peroxisomes. Before we even talk about what they do, arbitrarily on the model key from the manufacturer, which was a company called Staco, you look at these little gray skin circles with the purple interior. They call them peroxisomes. In the real world, lysosomes and peroxisomes are not color-coded. They're going to look alike. So the only way we can distinguish them off of a color-coded picture is with various types of chemical testing or really up-close analysis. So peroxisomes, they detoxify poisons in the liver. And the way they do, and the kidneys also, a lot of the old textbooks say liver only, but apparently the kidneys are also involved in some detox of poisons that we might find building up in our body, from bacterial infections, pharmaceuticals, you name it. So what they do is they produce hydrogen peroxide, H2O2. It's the same stuff you buy at the store, for antiseptic, for bleaching hair and stuff like that. Heck, 100% hydrogen peroxide is rocket fuel. 40% hydrogen peroxide is the max concentration they'll ship. Otherwise, the stuff might explode while it's being shipped. Very chemically reactive, very dangerous chemical compound when you get right down to it. H2O2 is an oxidizer. It attacks and destroys anything around it. So it's kind of like the Chronicles of Riddick. They had this evil dude, but they had to bring in the eviler dude, Riddick, to kill the evil dude. The first evil dude. So our cells might have poisons in them from a toxic mushroom, from a bacterial infection, some pharmaceutical compound that's really poisonous by itself as a side effect, even though it cures this problem we have and might make us better. Peroxisomes will attack that chemical with H2O2. They'll chemically oxidize it, neutralize its toxicity, and then we pee it out, poop it out. Well, mainly pee it out. So among other things, peroxisomes also perform beta oxidation of lipids or fats. That bacon, double cheeseburger burger that I keep talking about. Now you will hear me talk about this bacon double cheeseburger endlessly because I haven't eaten this morning and that's what's on my mind as soon as I'm done talking here. I have to get the kid from school first. The bun gets digested in the mouth and stomach, goes right into the bloodstream as sugar. The lean part of the meat, cheese, and the bacon, that gets digested in the stomach, a little bit in the small intestine, goes directly into the blood. But the fat content of the hamburger, the cholesterol in the cheese, the fat part of the bacon, that really gets digested in the small intestine primarily, goes into the lymph system. It does not go where everything else does. Ends up at the liver ultimately and some other cells and some other organs, and peroxisomes do the bulk of the initial fat breakdown to convert it into stuff that we can really get body fuel and energy from. And then the keto bros, of course, know all this. Okay, so mitochondria, weird little things. Well, heck, so are peroxisomes. They're also weird. They have their very own DNA, not a whole lot, mind you, just a tiny little bit, but it is their own DNA that's very different from ours. Mitochondrial DNA only goes from mother to offspring. Fathers, guys, we cannot pass our mitochondrial DNA on to our children because sperm cells really don't have a whole lot of mitochondria that's transmittable. So we have this maternal mitochondrial inheritance here. That was the basis of a video game years ago. I know it wasn't Mitochondrial Eve, but it was related to something like that. It was like a plague, pandemic, zombie virus type thing. I don't know if it's first person shooter or what. It's been so long, I've forgotten more game franchises that I've played than I can remember. Anyway, mitochondrial disorders, very debilitating. You do not want to have to inherit those. Okay, so we have some nice colorized transmission electron micrographs of mitochondria with all their internal layered structures, very up close and personal here. Talk more about these in physiology class when we cover cell respiration. Now, cytoskeleton elements. You don't think of a cell as having bones, and they don't, not in the sense that we do, but they do have a skeleton, and it's made out of proteins. So these proteins are thread-like structures, and there's three categories based on size and chemical structure. Microtubules are one. Microfilaments are kind of the smallest. Microtubules are the biggest. Microfilaments are the smallest, and intermediate filaments, as the name implies, are intermediate in between the two in size. Microfilaments being the little guys, they're basically made of round ball-shaped proteins that go together like pop beads. And if you ever played with pop beads, I need to order a ton of them off of eBay to make chromosome models for teaching molecular biology. They go together like these strings of beads, and you pull on them and they're tough. They don't come apart easily. So we have these little round balls of protein called G for globular actin. They stick onto each other just like the pop beads, and make these strings called F actin or filament. Let me tranquilize somebody's audio here. Who do I hear besides? There we go. Mute everybody. All right, so that's that for microfilaments. Microtubules, way bigger by comparison. Those are big hollow tubes, and they're made of a spherical protein called tubulin as well. So think of a string of beads that's coiled up around a fingertip like a slinky. When I was a tiny little kid, I think I got into my mom's or aunt's costume jewelry, and they had a big old pearl or malachite necklace, and I was just a little 5-year-old kid just playing with this thing. I wrapped it around my fingertip, made a coiled tube out of it, broke it. Holy guacamole balls, did I catch hell for that. Public service announcement to any 5- and 6-year-old boys out there: do not find your mama's jewelry box and break everything in there. It will end badly for you. Anyway, so that's sort of how these microtubules go together there. When you have a combination of two different building blocks, it's called a dimer, di meaning two. So you take alpha and beta to form a dimer, and then you string these together and coil them up, and you get this tube-shaped structure. Okay, intermediate filaments. These are different. They're called intermediate filaments because they're intermediate in size between microfilaments and microtubules. And the proteins are shaped like dumbbells. They have the weights at the end here, the big parts at the end, and then you have the skinny little part you wrap your hand around when you're curling dumbbells in the center, or maybe barbell shaped if the shaft here is really long. Okay, so the way these go together is as you see, and the actual physical texture, the closest real world analogous thing is string cheese. That's sort of like the physical texture of an intermediate filament. Okay, so if you're gonna see these, you gotta have the big kid staining techniques, fluorescent staining procedures. Some of these fluorescent dyes are kind of ever so slightly poisonous. I have a jar of something called fluoroglucinol around, and oh my God, the training and stuff I had to go through just to be authorized to use that in the lab setting was extensive. At the time, I complained about it. Now it's nothing compared to some of the training we have to go through. So there's something called immunofluoroscopy. The different characteristics of these proteins chemically will bind to different chemical dyes or different chemicals, ligands, and then we add the different dyes that light up under a black light: blue, green, red, orange, purple. This is a big cell here, probably a big neuron from the brain or spinal cord. And they applied 3 different stains to this. And then they put it under the microscope and used a UV light, a black light, for the light source. Very costly, very time consuming, and the chemicals to get this are a tad on the hazardous side. So in this view, the microfilaments stain red, intermediate filaments show up as green, And the big microtubules on this image show up as blue. And whoever made this was very courteous. They put a size marker, 5 microns. So this little bar down here denotes a length of 5 microns, 5 millionths of a meter. So 5, 10, 15, 20. This is a big cell, at least 100 microns across. Okay, we're coming towards the end here. We have organelles for movement, locomotor organelles. Now, these are microtubule-based structures. So you have what are called centrioles, and then a version of that called basal bodies. In this cell model that we have, both old and new versions, they have a pair of centrioles at 90-degree angles to each other. And these structures, that damn dog wants out again. These structures are called centrosomes. So a centrosome is a pair of 2 centrioles here. And these act like little molecule-sized motors that actually cause movement of long, skinny structures made of microtubules. So centrioles and basal bodies, you're going to have a radial arrangement of microtubules like 1, 2, 3, 4, 5, 6, 7, 8, 9 on the outside, and then 2 to 3. So you have a 9 plus 2 or 9 plus 3 arrangement in the center. The basal body occurs inside the cell and sort of where the movement begins. And then the part that sticks outside of the cell is called an axoneme. There's actually a motor molecule called dynein that changes shape when you add ATP, body fuel energy, cell energy to it, that generates that movement. Okay, so this is a SEM scanning electron micrograph of cilia. Cilia are one type of locomotor organelle powered by these basal bodies, and they just whip back and forth to generate movement. You'll find them on what we call ciliated epithelium in the upper respiratory tract. Basically, they help clear particulate matter, help move dust, specks of dust, rock, whatever, away from the lungs. Now, flagella are large whip-like structures that sort of whip around back and forth. These are only found singly, one at a time where they occur, whereas cilia form like vast, I don't want to call it forests, but you got a lot of cilia. Flagella are only found on sperm cells, which are male reproductive cells in the human species. Yeah, this is kind of like a really fancy, hush, dog, I'm almost done. Somebody needs to go out and pee and poo. You know that 14-year-old geriatric dog I'm always complaining about? Well, that's her. So the way flagella and cilia move is the flagella sort of whips around and you have like a propeller-like. Ever take a rope and twirl it? I know guys don't do this, but we had jump ropes and every now and then we try to twirl them out of boredom. And that's sort of the movement you get. Whereas cilia is kind of like a whipping, just a simpler back and forth movement. Okay, the basal bodies for flagellum and cilia are located inside the cell, in the cytoplasm, and then we have this yellow line here, this cell membrane. And then the axoneme, again, is the part that projects outside of the cell. Now, there's a third category of movement with its own organelle, and that organelle is called the pseudopodium. They form and then they dissociate and go into nothingness again. So we say they're transient structures, they're not permanent. They form in a matter of seconds, they disappear in a matter of seconds. And these are used in what we call amoeboid movement, where the cell changes shape, relocates its internal organelles and cytoskeleton elements in order to move. So the cells that can do this, they transition between two states, a solid gel state like jello, jelly, jam, and then a sol state, which is more liquid that flows more like water. if you and I could do that, we would be like the Terminator in Terminator 2, that guy that would turn into liquid metal and flow under doors and then reform on the other side. I so wish I had that as a superpower. I'd never have to worry about carrying keys around again. Like I show up on campus to teach and I don't have my keys. I don't have to beg the dean or department chair or the front office. I just dissolve myself into a puddle, flow underneath the door, reform on the other side, and let everyone in from that side. That would be so awesome. Okay, so in other terms would be plasma gel for gel state and plasma sol for sol state. Now, ordinarily, we would only think of predatory pond scum like amoeba that move like this, but in actuality, a huge number of human cells, quite possibly most of them, can move like this at some point. White blood cells, which you call leukocytes, I like to call them WBCs. Red blood cells, erythrocytes or RBCs, are able to do this. Neurons, quite a few neurons and what we call glial cells can do this as well. Okay, so that is up for cell. Oh, that's it for cell ultrastructure. Before we leave, I need to go here. And now I need to stop share and go to the new share here real quick. This is our Canvas shell for the lecture schedule. This lecture is for module number 2, and I have an amoeboid movement video here. I'm going to post this video when I'm done recording, but you can totally see this. So this is a 30-second YouTube video. You can see the solution state here. The cell has just turned itself into jelly, and it's pulling back some of this old material here, and it's flowing into new areas here. And then somewhere you see this rigid part of the cell that doesn't change. That's the gel state or plasma gel state. So this here's an amoeba, which eats littler creatures yet in dirty swimming pools, rivers, and lakes, and sewer water and stuff like that. But some of our cells, our immune system cells, can actually take this form and move like this. Okay, I have to stop share. I have to go get my kid and let that annoying dog out. So let me stop recording here, and there we go. Hold on.